Good afternoon. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of the Biochemistry Focus webinar series. Topics in the series will include research areas in a variety of molecular bioscience areas, as well as practical sessions on career development. Each webinar will give you the opportunity to ask questions of experts via text, and we welcome suggestions for future topics that will be valuable to you and speakers who could address them. Please go to the Biochemistry Society website for more details. I'm John Colyer. I'm an academic from the University of Leeds with a chair in biotechnology, and I'm hosting this webinar because I lead a program in biopharmaceutical development at the master's level. I'm delighted to introduce our speaker this afternoon, who is Siân Estdale. She is the head of scientific affairs, clinical trial testing solutions at Covance Laboratories. And Siân will be speaking this afternoon on a brief history of biopharmaceuticals, the scientific drivers for the adoption of large molecule therapeutics. In this webinar, Siân is going to present a brief historical context of large molecule drugs, the current market for approved drugs, the modalities and the therapeutic targets that they address. She will go on to explore the mechanism of action of some of the blockbusters in this field and compare them with small molecule drugs, highlighting the major differences, and finally discuss the drug development pathway and the probability of success. As with all these seminars, you will have a chance to pose questions at the end of the webinar. You can draft those questions in the chat box to the side of this during the talk, and they will be fed through to me to put to Siân. So I'd like to now welcome Siân Estdale to address us on biopharmaceutical development.
Lovely. Thanks, John. I'm just going to show my screen, which hopefully will appear. Hi, everybody. It's really nice to be invited to give this talk. I have worked in the field of biopharmaceuticals now for 20 years, and I just wanted to give you an overview and give you a flavour of biopharmaceuticals and hopefully get you excited and interested in them as modalities for the new therapies. So John's outlined my talk, and let's just jump straight in. This is the best sellers list. It's something that I keep up to date. Every year I buy Le Marie, which is an industry magazine that gives a record of what sold best the previous year. So I just keep a running tally. So you're thinking about biopharmaceuticals. Why are they such big business? Because they bring in an incredible amount of money. Here you can see I've tracked, and this is all I can fit on, from 2012 to 2019. And cancer antibodies made nearly $60 billion alone last year. Anti-inflammatory antibodies made $43 billion. And then anti-TNF antibodies make $37.7 billion. They are huge money earners. You know, the total for this selection is about $223 billion. Now, this tells a story of itself. Back in the day when antibodies were being developed as targets, they were really helpful because they actually utilized the body's mechanisms to interfere with therapeutic progress of disease. And you can see that back in 2012 through 2016 and before that, actually, the #1 antibodies were TNF. And these were one of the original targets. So it's an anti-inflammatory antibody that interferes with quite a lot of disease states. And then you can see that the other anti-inflammatory antibodies that were then subsequently produced are now #2. And #1 is the cancer antibodies. I'm going to talk a little bit more about the checkpoint inhibitors that were approved only a couple of years ago, and they've made massive strides for the biopharmaceutical industry in that they offer a completely different way of ameliorating disease states, and they can be used in combination with these anti-inflammatory antibodies, which again underscores why they're such successful therapeutics. So that's just a snapshot of why biopharmaceuticals are big business, because they bring in an awful lot of money. Let's just backtrack a little bit and think about what biopharmaceuticals actually are. So what they're not is biological medicines. So biological medicines are mechanical products. So they're not, you don't manufacture them per se, but they are harvested from living organisms and tissues. And they're not synthetic molecules. So we can't make them in the lab. We can't go into an organic chemistry lab and create these. I mean, we're getting better at it. Don't get me wrong. We can create peptides now up to quite a large number of amino acids. But the thing that separates biopharmaceuticals is the fact that we use recombinant DNA technology. And what we've learned to do is get that genetic information into the cells such that we can make a protein of choice. And there are several modalities that I'll touch on today. Monoclonal antibodies, of course, being one of them. As you can see, they're big business. Recombinant proteins and peptides, therapeutic vaccines, which I'll come to at the end of my talk, and also cell and gene therapies. And we've seen a lot of interest in this regenerative medicine and stem cells, particularly, over the last 10, 20 years. So because we make these products from cells, we have a completely different manufacturing process. And what I'm showing you here is what that looks like. These are massive bioreactors. And if you've never seen them in real life, when you get to the stage of needing to manufacture for a commercial product and you're having to produce a lot of these, they're massive bioreactors, the size of a two-story house, where you have to, walk up the outside in order to be able to access adding things or taking waste away from the bioreactors. And this is sort of very much old school at the moment. These manufacturing processes obviously still exist because they represent massive investments within the industry to create something that will manufacture proteins from these cells, but they are being replaced increasingly by this disposable technology, which was a remarkable innovation, really, because rather than having these huge metal structures, which then needed to be cleaned out before that you went into a new manufacturing process, the disposable technology actually places a plastic bag inside the bioreactor. So it's disposable, there's far less downtime, you don't have the same cleaning regime, and they offered a new way for us to develop further our capabilities in manufacturing and make it accessible more broadly across the industry. So let's take a look at what specifically some of these blockbusters are that we mentioned straight away. The #1 bestseller, and it has been for a number of years, is adalimumab, and I'm sure that you've heard of it, or by its trade name, which is Humira. This is the anti-TNF alpha antibody. So it's an antagonist, and it's fully human, and it made sales of $20.5 billion in 2018. It's gone down slightly to $19.7 billion in 2019, and that had been going up year upon year upon year. Why is it changing? Because of biosimilars, and maybe we'll come back to that in a little bit. Let me pick up on the checkpoint inhibitors, because this is pembrolizumab and nivolumab, Keytruda and Opdivo. So these were the checkpoint inhibitors that were approved not too long ago, made by Merck and Bristol Myers Squibb, and in combination these represent a couple of the anti-cancer antibodies, and they have, together with others, encouraged this leap of the anti-cancer antibodies up to becoming #1 because of the way that they can interact with a huge number of different cancer pathologies. And then, of course, there are the anti-VEGFs. So these tend to be needed in the progression of some disease states and some cancers. They stop the development of additional blood to the tumors, and in these other disease states. And so the anti-VEGFs have also been, very popular. And these represent the blockbuster biopharmaceuticals for this last year. And really, that hasn't changed very much in the last 10 years or so. The new ones that we see coming in are these checkpoint inhibitors. And also these other, which are variants of monoclonal antibodies, and we call them fusion proteins. They incorporate the specificity of the monoclonal antibody, and they also have some of the features of a monoclonal antibody, but they're just created slightly differently. So let's look at why these molecules are making the news. And what I'm showing you here is the checkpoint inhibitor Keytruda. So it's a monoclonal antibody, and one of the attractions of biotherapeutics is the fact that we are, as I said earlier, utilizing the body's own mechanisms for interfering with disease states. And within cancer, we have found that within certain patients, it's not every patient by every means, the tumor will express a ligand which binds to the patrolling T cells or NK cells, that when the T cell or NK cell docks, it sends a signal deactivating the cell so that the tumor cell is not recognized by the immune system. And what we found is that when you've got an anti-PD-1 monoclonal antibody, it binds to that ligand on the T cell. Therefore, the immune cell is not able to send a signal to deactivate the T cell. And you can see some astonishing results. So if I click on, I know these pictures are just amazing, aren't they? This is a guy with melanoma. And he's refractory, which meant that the standard of care, which was ipilimumab, didn't work for him. And his melanoma was at a severe stage. And after only 3 cycles of treatment, you can see that the melanoma is receding and receding, because what's happening is that his T cells are able to identify the melanoma cells and able to attack those tumor cells. So they have quite dramatic impact on certain patients. Again, it's not every patient, and that brings with it an additional challenge about how do you identify the patients that are going to be impacted by certain therapies. So in terms of the drug development pathway, there's a recent paper that came out this year, and we have now new details on how long and how much it costs to develop a new biopharmaceutical drug. And it's gone up to 3.1 billion. Now, that isn't for a single product, but it recognizes that this is big business. It takes a huge amount of investment. So for 1,000 or thousands of potential new medicines, only one of these will get to market. And this bar across the middle represents the different stages of the drug development process. It's sort of represented in a linear fashion. And of course, it never goes like that. You will make steps in a certain direction, trying to understand the mechanism of action of your disease state, trying to find a therapy that interacts. And there may be several modifications and different processes, which mean that you go from one stage back into the research stage, and you might progress and then find other factors that need to be amended. But it is an astonishing number, this 3.1 billion. And on average, these biopharmaceuticals are not cheap. They typically cost about $100,000 per treatment. So that's not a single treatment, but it is a huge amount of money, and why industry is getting more and more into this. There are now over 50% of all therapeutics that are being developed are biopharmaceuticals. On average, it takes 10 to 15 years. However, that's being challenged very much so by the recent development of gene and cell therapies, and by also new biosimilars. We have seen curtailment of that drug development timeline, sometimes down to 4 to 5 years, which is remarkable given the amount of data that you need to show to the regulators to be able to take your product to the commercialization stage. So all of this drug development pathway is regulated. There are requirements to demonstrate safety of your product, efficacy of your product, and also quality. So that's down to the manufacturing that we just sort of touched on a couple of slides before. You've got to show that your product that you manufactured is stable and it's consistent. So lot-to-lot manufacturing is going to produce exactly the same impact that you've demonstrated through your preclinical or your safety trials into your clinical, which are mainly your efficacy trials. So why are they doing so well? The specificity to the target is so precise and so exquisite that compared to small molecules, they have far less of target toxicity. And that's one of the challenges with small molecules, that it's usually the liver and the kidneys that are responsible for processing those small molecules. And that's why we tend to have animal studies so that we can assess that. So that specificity is really key in terms of why biopharmaceuticals are positive targets when you're considering how to impact a disease state. Also, the probability of success compared to small molecules is typically higher. And there are a variety of reasons for that. One of them is because of its specificity. And also the IP landscape is much more complex. So from a small molecule pathway, you can synthesize that molecule in the laboratory and you can readily understand the molecule. It's a single molecule type that you're looking at typically. And that molecule that you can target and that you can also analyze accurately and specifically is what has the therapeutic impact. It also means that somebody can come along and reverse engineer that synthetic pathway and create the same molecule. And they can use the data that you've generated from a safety and efficacy perspective. And when your IP when the patent has expired, then that company can come in and steal some of your market. And you noticed in the previous slide when we were talking about what were the top blockbusters and why adalimumab was making less money, and I said it was to do with biosimilars, that is actually what's occurred here. So from a biosimilars perspective, it's a much more challenging pathway to be able to reverse engineer the amino acid sequence, or to reverse engineer the DNA that would give rise to that amino acid sequence. And also because you're not using the same cell line, necessarily you may produce the same amino acid sequence, but the post-translational modifications that are a result of that specific cell line might be different. And the thing that you have to show when you're developing a biosimilar is that there are no changes in efficacy. So there is far less threat from the production of biosimilars, even though we see huge amounts of activity within that market these days. And this is what led to the increased likelihood of people turning to biopharmaceuticals. Here is a graph that shows the probability of success, which comes from the Centre for Medicine's research. And you'll see that in every part of the pathway up to submission to market, that the biotechnology products, which are in the dark blue bars, do outperform the small molecule entity comparators. So again, this is pivotal information when you're thinking about what sort of pathway to go down in terms of your drug development. So I put in this slide because I thought it was interesting to see that, large molecules have only been out for a short time. They are dependent on this technique that was developed by Boyer and Cohen, which is the recombinant DNA technique, which enables us to produce proteins of choice from a cell line. And Eli Lilly were the very first company with Genentech to produce insulin by recombinant technique. And then you will see this, if you look closely, this rapid development of certain modalities. So, we had vaccines back in the 90s. And we had our very first successful gene therapy in 1990. But if you look at the bottom here, you know, in the 90s, in the 10s and the 2020s, it's called the noughties, isn't it? We have this rapid utilization of monoclonal antibodies as target molecules to interfere with therapeutic disease states. So we've gone from 7 to 16, and last year, at the end of 2019, I added up all of the molecules that had been approved, and we had 68. So you see this rapid acceleration. Gene therapy is also big news, and we have seen huge amounts of interest in that, where we had our first successful gene therapy quite a while ago, but the completion of the Human Genome Project has actually you know, given an injection of information that we've begin to utilize as we understand the potential for gene therapies. They have been thwarted by lots of challenges, which I will touch on in a little bit, in probably the same way that monoclonal antibodies have. There have been challenges in terms of their manufacture and in terms of the interactions that were caused that were not always positive with certain patient groups. So it's been a voyage of understanding how to produce safe and effective products. Already this year, we have seen 2 gene therapies approved and 8 monoclonal antibodies. So it definitely looks like that trend is going to continue throughout this decade. And you know, it'll be interesting to see what happens beyond that. You'll see that I'm calling out COVID-19, and at the end of the talk, I will touch on the impact that that is having on the industry as well. So I hope that gives you a snapshot of the way that the biopharma industry has developed over the last 50 years. So this talk was to look at what are the scientific drivers for developing what we call large molecules of biopharmaceuticals. And I thought it was worth touching on the fact that there are these positives. So in terms of molecular weight, then biologics are much, much larger. The typical example I use is that for small molecules like aspirin, it's like comparing a Rubik's Cube. And if you're comparing that to a monoclonal antibody, it's like comparing that to an elephant. So a Rubik's Cube to an elephant gives you that representation of the changes in size, also in terms of the degrees of freedom, also the degree of which we can accurately analyze and understand the new molecule. In terms of selectivity, we've already called that out. You know, it's much more selective because we're using a biological process and we have less off-target impact. Species selectivity can be challenging, and it is something that has given headlines within the news when, you know, there have been failings with some biopharmaceutical products, because sometimes the animal species does not reflect the mechanism of action. So, you know, that can be challenging. One of the positives about large molecules is that they tend to be, they have long half-lives. Like, you know, from taking medicines off the shelf from your pharmacist, that you typically have to take them, you know, maybe 1 or 2 a day, every 4 hours. Whereas for large molecules, they typically last a long time, many days. And some of the monoclonal antibodies, you may only need to have injections, you know, once a month. In terms of distribution, though, the small molecules, it's extensive, which is what can lead to this off-target effects that you don't want, whereas for biologics it is restricted. So, you know, you tend to be dependent on the vascular system to distribute that molecule. However, one of the major things that we have to be really aware of is immunogenicity. So for small molecules, there's hardly any instance of immunogenicity. So we're not activating the body towards that therapeutic such that it ameliorates the impact of it. Whereas for biologics, it's quite a common challenge, and it's something the regulators are really hot on, and it's something that we have to demonstrate thoroughly, both through the non-clinical stage and the clinical stages, to look at the incidence of how somebody's immune system is alerted to this new therapeutic. I've put this in because it shows the harmony that can exist between large molecules and small molecules. One of the great things about large molecules is the specificity. One of the great things about small molecules is its toxicity. And antibody-drug conjugates, or ADCs, actually represent the best of both worlds. Now, they have been through many different phases of development in terms of finding the best way to get the exact receptor that will be internalized and the right toxin that doesn't find its way into the circulating system but only gets released when it's internalized into the cell. And what I'm trying to demonstrate here is the mechanism of action. So the first point is through the circulating system that the antibody-drug conjugate, by virtue of its monoclonal antibody specificity, binds to the antigen on the cell surface. And then if you've selected the right receptor, that then gets internalized. So the whole molecule gets internalized. Through lysosomal degradation, then the toxin is released into the cell cytosol, resulting in killing of the cell. And I thought it was worth showing this real life example of a guy, and it was in the news, but I was at a conference where the medic who actually administered the drug to this guy was telling the story, and I thought it was a great story. So I had the permission to also show this slide, which on the left side shows a guy who has lymphoma. And he's double refractory to the standard of treatment, and essentially he was unable to be treated any further. Christie was given some new antibody-drug conjugate by Seattle Genetics, which are the company that made it. And he agreed to take part and came in, and they administered it through an IV over a period of time, maybe an hour, 2 hours, and sent him away. And he came back the next day feeling awful, and they just made him comfortable and gave him, a paracetamol and put an IV drip in, and looked after him for a little while and then sent him home. And this is a remarkable story because what happened is that the specificity of this monoclonal antibody drug conjugate had sought out all of the tumor lymphomas that were expressing this receptor, and the toxin had killed all of the cells and lysed them, and what he was suffering was tumor lysis syndrome. So it was so effective that during the process they were really worried that he was going to be negatively impacted, but in the end it was an amazing story of success. So gene therapies actually have been theoretical for quite a long time. We have thought about how to regulate or interfere with, silence some gene, or maybe even replace and repair it, and thereby offer a cure for a disease state. So rather than dealing with the symptoms and trying to ameliorate the symptoms, and very rarely coming across a cure, frequently these are chronic administration of a drug, gene therapy actually offers the potential for a cure. And within industry, they call it one and done. And the very first gene therapy was approved in August 2017. And this is a schematic that one of the guys in my lab created to show the idea of how we can interfere with these different processes. They also highlight the selection of ways in which we would use viruses potentially, or polymers or dendrimers, to be able to get that information into and target it into a cell. Very often we choose viruses because through evolution they have this incredible way of penetrating a cell to deliver genetic material. And one of the difficulties with large molecules is that they cannot penetrate the cell. They can only dock onto the outside of it and interact with a receptor. Whereas these gene therapies, by virtue of harnessing this, what nature has given us, enables us to find different ways of getting that genetic information into the cell to interact in a variety of different ways. The challenges of gene therapies are that we have seen some undesired clinical outcomes. There have been some deaths which have made the standard of, proof of safety and efficacy that much more difficult to demonstrate, because naturally, everybody gets a little bit nervous when we don't understand the impact that we're having on a patient's immune system particularly. So we have seen off-target effects, cytotoxicity. One of the reasons for selecting certain viruses early on was that ability to penetrate the cell, but sometimes, even though the best efforts were made to make these viruses non-transmissible, they still saw some of that happening. So, there have been different techniques developed to manufacture viruses that aren't going to be transmissible. We've seen some difficulty with impurities. And also, sometimes the viral vector that we have chosen, some of the patients have already been exposed to, they will already have an immune response to, and that therefore interferes with the impact of the viral vector that's trying to deliver that genetic material, because it immediately gets recognized. And then alongside gene therapies, we've got cell therapies, and I've put in a link to a video which I don't propose to show, but it is an amazing story of how these CAR T, which is the chimeric antigen receptor T cells, were created, and they utilized a virus. It was the HIV virus, which they made sure was non-replicating. And ex vivo, they removed target cells from the patient. And then outside of the body, they introduced the virus with the genetic material such that it used the mechanisms within the cell to express a receptor. And then they were grown up and put back into the patient. And so there are a variety of different cancer therapies that have been tried, and they've obviously had a huge amount of interest as well. And this is a schematic which just explains what I was talking about. And these are a very different, I'm sure you'll recognize, manufacturing process to the one that I described earlier, because you're actually manufacturing using the patient's own cells. And that means, from a drug development perspective, a much different and more challenging way of assuring, safety and quality of these products before they go back into the patient. And sometimes, we're thinking about what's the risk, and what's the impact going to be, and sometimes you might not have all the answers to that, but the benefits outweigh the risks, and so you will go forward anyway. So there are a number of other things that we consider when we talk about cell therapies, how in terms of safety and efficacy. How do we identify the T cells, and does it matter? What if there are other cells, or how do we remove them? And how long is that gene going to last? Is it going to be transient, or is it going to be incorporated into the DNA, which isn't positive always? So how long is it going to last? And, as we were identifying, what is the risk to the patient? So finally, I'm coming on to talk about vaccines, which are another modality that I highlighted right at the beginning. And the reason that I've left it till last is because I obviously have to mention COVID, which has had an incredible impact on all of us. Here we are having virtual presentations, which are great, but we've all had to adapt to this new scenario. So vaccines have obviously been a very interesting path to take. They are not chronic by nature. Industry has a very different view of them. And up till recently, I think I would argue that governments have had a very different view of vaccines. They are not seen as big money earners. We tend to target the ones that are going to have the most impact on our population. And because they're proactive, so you're trying to give somebody immunity before they get exposed to a pathogen. You don't always recognize the financial value of those vaccines, and there are lots of different types that we have looked at over the years, depending on when they were created. So, we can have the live attenuated virus, so it's lost its pathogenicity, so it can't create disease. But if you inoculate somebody, then their immune system has an opportunity to develop an immune response to it. We can inactivate them, or if we use our recombinant technology, we can have subunit vector and DNA vaccines. And the reason that I'm talking about these all is because one of the things I wanted to highlight was what is industry's response to COVID-19, because you've seen governments all over the world talking about the need for a vaccine so that we can get out of lockdown, and being willing to spend huge amounts of money. And you have seen big pharma in the news working with anybody who can produce something that might be successful. So I thought it was worth just highlighting what the mechanism of action was for COVID-19, and what that brings out is the potential for targets that we could, within industry, go for as therapies for COVID-19. So you will have heard about the S protein, which binds to the ACE2 receptor on a cell, and I think this is a lung cell. So the virus gets internalized by endocytosis, and then because of that, the RNA gets released into the cytoplasm following uncoating of the virus, and then the viral RNA gets translated. So there are a number of transcription events that need to occur so that you can get the proteins and also the genomic RNA that then self-assemble into a viral capsid so that it can be released out into the extracellular space outside of the cell. So these have represented lots of targets. Can we prevent this docking, first of all, and what sort of molecules could that be? Would that be a monoclonal antibody? Can we interfere by getting RNA into the cell in a complementary way, such that some of this information for creating the virus cannot be produced, and therefore you can't get this spontaneous assembly of the virus, therefore inhibiting the pathway? And there have been a huge amount of interest. So I have looked at the treatment that's out there. So you can either block entry, block replication, stimulate the innate immune response, or enhance the antiviral innate immunity that we have. And there are a huge number of vaccines that have been produced. And I've put this link on here, which takes you to the WHO website, and I've got it here, just to illustrate to you pharma's response, because we have a number of pages, and I am just going to flick through these, hopefully to show you that we have VLPs, which are vaccine-like particles. We've got non-replicating viral vectors. We've got protein subunits. We've got RNAs. We've got, again, vaccine-like particles, and I flipped over the first two. We've got inactivated products, and we've got non-replicated viral vectors. And you will see the industry that you will have heard of, Oxford, AstraZeneca, the Beijing Institutes, Gamaleya Research Institute, Janssen. It has been a monumental effort on behalf of, the large molecule biopharmaceutical industry to try and come up with a vaccine that will hopefully get us out of our present situation. So I thought that was fascinating, and I wanted to share that information with you. And so I'm just going to wrap up my talk and open it up for questions, because in summary, biopharmaceuticals are a hugely important growth driver for the pharma industry. You saw by virtue of those numbers that it can mean big business. They are incredibly expensive to make, and they are incredibly expensive to administer. So these are not a cheap option, but you've seen some of the impact that these molecules can have, and this is why they continue to be developed. They're less toxic than small molecules, and they circulate for longer, but they can elicit immunogenicity, so your body can be highlighted to them. They do have a dramatic impact on the quality of life for some patients. And for the first time, we are potentially offering a cure through gene and cell therapy. So rather than chronic administration, we are looking for our industry to produce that one and done, to genetically modify. And as you've seen, there's been a huge response from the biopharma. So, is this the industry that's going to get us out of COVID lockdown? My fingers are crossed, even though I know how long this drug development process takes. People are talking about rapid production of these vaccines. So that would be amazing. Thank you for your attention. I am more than happy to answer any questions. Thank you.
Siân, thank you so much for a fascinating canter through
yeah, it was, wasn't it?
the history and the achievements of the biopharmaceutical industry. So questions are coming in from the audience, and can I ask them to keep sending them? I will pick up questions and try and group them together so that they make good sense. So you mentioned a couple of times in your talk that the large molecule drugs were more successful than small molecules. Can you just summarize for us why that is?
I think it's because of the specificity of the molecule. So the drug development pathway arose out of the need to demonstrate safety in animals before you administered to humans. And because we are replicating the body and the way that it interacts, our bodies are used to proteins. And so they know how to process them, they know how to degrade them, whereas small molecules are alien to our bodies. And that's why things like paracetamol are toxic. Not because our bodies can't clear them, because eventually the liver will degrade them, but it's just a more complex process. So I think it's because we're able to understand the mechanism of action perhaps more readily and how it interacts with the rest of the body, and that's what leads to the success. But I'm sure there are other factors. Go on.
So if I can follow up with a question from the audience that wants to explore how these drugs can get to site of action that matters. You were illustrating it with extracellular receptors or targets for the molecules. The question really wants to explore the blood-brain barrier and how do we overcome delivery beyond the blood-brain barrier, and also, of course, the plasma membrane barrier, and how do we access intracellular targets. So what's being done there? What are the prospects?
It is a fascinating area, and you're right, there's a lot of interest, especially in the blood-brain barrier when we're looking at dealing with neurodegenerative diseases. And there are monoclonal antibodies that are being created for that. It is a challenge because evolution has created barriers in this way to protect us. And yes, we use usually the circulating system. So we, administer through IV because we can't take large molecules in through the oral route because we have digestive enzymes. And it is a route that is being looked at, but there are also many other barriers of safety to recognize when we're looking at alternative routes of administration. So I'm not sure that I can give you a particularly precise answer, except to say, yes, they are being looked at, and there is limited routes for success. They tend, I have heard about people using targeted therapies for solid state tumors. These are one of the ways that industry is very interested. We're very good at looking at blood tumors, but solid state tumors actually offer more of a challenge, purely because how do you get your circulating drug to that environment? And so they're looking at ways to either through administering locally, like injecting locally by visualizing a tumor where to administer the drug, or by other mechanisms which can, attract the circulating product to that site to be of use. Yeah, all I can tell you is that it's a huge area of interest, and hopefully we will get to a point which is both safe and efficacious, and we can start, having success in other areas.
So
I hope that answers it.
Well, it begins to, doesn't it? So, and there's a further question that follows on, exploring smaller large molecules. So antibody fragments, single chain variants of antibodies, non-antibody versions, nanobodies, and other very small binding partners. What are the issues that they face?
Well, the larger the molecule, typically the longer that it's going to last within circulation. So when it comes to smaller peptides that maybe have the working part of the molecule, maybe it's the variable region. The thing is that they get cleared fairly rapidly. So what tends to happen is that you try and find other mechanisms to make it evade, removal from the circulating system, such it's got more opportunity to have a therapeutic effect. And either you add something like a PEG molecule, because that's traditionally used to extend the half-life. It's used in one of the fragments, which is an anti-TNF, that's got a PEG molecule on it, or you tend to conjugate it to something like the Fc portion of the monoclonal antibody. That has a function of interacting with the endothelial cells while it's circulating, and they prolong the half-life because it gets taken into the cell by endocytosis and then released when there's a pH change. So we tend to want to have the benefits of the large molecules with the specificity of the half-life, alongside it. Having said that, one of the massive benefits of targeting smaller peptides is that you no longer have to manufacture within a cell and all the challenges that go along with that. And theoretically, perhaps you can synthesize it in the lab. And as I touched on at the beginning, we are getting much better at being able to synthesize amino acid sequences of extended length.
And then I have one more question on protein chemistry, so raised from the audience. So the observation is that most therapeutic monoclonal antibodies are of a particular class, IgG1, whereas Keytruda is an IgG4. So what are the characteristics of these different classes that are useful or problematic?
Yes, excellent question. So one of the evolutionary advantages of monoclonal antibodies is that whilst they've got this functional ability, which changes depending on the antigen that it's directed towards, which is in the Fab region, the Fc region is conserved through evolution. And there is a significant difference between IgG1s, which the person who asked the question quite rightly said most monoclonal antibodies are, as compared to these checkpoint inhibitors, which are IgG4s. And the Fc region is made to interact with the immune system. So I already described the ability for it to interact with the endothelial cells, and it's via the receptacle, the FcRn. And this is the neonatal receptor, which it prolongs the half-life of these monoclonal antibodies. But it has a number of other functions which causes it to interact with circulating T cells, NK cells. Now, that can be a positive interaction when you are thinking about your mechanism of action. Perhaps you want to, target a cancer cell, and then you also want it to be recognized that you've interacted by linking onto the Fc, and that T cell can then engage in destroying that tumor cell. But sometimes you don't want that to happen. So the IgG4s in this case, which are the checkpoint inhibitors, were made to block that interaction on the T cell, but you don't want engagement with other NK cells, which would result in wiping out these T cells. So it's the mechanism of action that you're wanting to elicit that gives you the idea about which, IgG1, IgG2, or IgG4 that you want to engage with.
And following up on that, a further question that's just popped in: do they always have to be humanized? Are any therapeutics not of a human architecture?
There are chimeras out there, and so the history goes that we started to create monoclonal antibodies from Fab regions that were created in mice, but they were recognized as mouse, and so what happens is that we learnt to create more human versions. There was also the problem because most of the cells that we choose are mammalian in the manufacturing process, because we want it to create a molecule that undergoes similar post-translational modifications. What happened in the early stages that we tended to use Chinese hamster ovary cells, and of course they did what mouse cells do, and they produced glycosylation that was important for mice, and therefore, what mice would recognize as self. Whereas in humans, we have very different glycosylation patterns. So we came up with humanized manufacturing cells, which produced a subset of glycans or glycosylations that were not recognized as mouse, but more humanized versions. Did I answer the question properly?
Yeah,
okay,
absolutely.
good.
So I'm going to switch to the viral side, the viral vector side of gene therapy that you introduced to us. So the question relates to the immunogenicity of viral vectors. So what do we need to do to overcome the immunogenicity that they pose?
So the immunogenicity that we might have impacts the people that are going to be administered to. So in theory, you want to be a one and done. You want to get that genetic material into the target. So the immunogenicity doesn't have the same profile that it does for these chronic administration. So if you've been exposed to this viral vector that is wanting to be used, then your body is already going to have antibodies to it and recognize it. So what's happened is that there are lots of really clever people there that look at the capsid proteins, and they try and represent them by a hybrid of different viral vectors, such that it becomes non-representative of, say, adenovirus 5 or adenovirus 7. They may be a combination of capsid proteins, which is what the body gets exposed to, which might be of 3, 5, 7, and 9, and you get this mosaic. So it's sort of an atypical virus, but it still has the advantages that evolution has given it, to be able to get into the cell.
But you're not primed to it. Is that the point of that word?
Yes, you can't be primed to it.
You're not primed to it.
Yes, thank you for that. Yeah.
Yeah, and if I can turn to questions that sort of haven't come from the audience, you mentioned that not all patients will benefit from a given therapy. How do we identify those who will when the therapy is so expensive?
Yeah, it is the $20 million question. And so one of the examples that I use is that of Herceptin. So there are various types of breast cancer, and they have a different expression of different receptors on the breast cell surface, and they get categorized as such. And when Herceptin came along, it was targeted towards HER2. So, and then overexpression of HER2 on the breast cell. Now, if all breast cancer patients had been treated with Herceptin, it would have failed, because it would never have shown, that it could help reduce those breast cancer tumors in that cross-population example. So what had to happen is that those patients had to be identified. So, they do a lumpectomy and they would analyze the cells, and for a proportion that were in that grade 3-4, so overexpression of HER2, they knew that they would do well on the trial. So they could put them into the trial, give them Herceptin, test them against their placebo group, and therefore you'd see this greater probability of success. Which brings us to this whole point of patient stratification. How do we recognize within a disease state the different population groups? And what are the factors that distinguish them? And if we can recognize the factors, what are they? Are they, genomic factors? Are they immunologically based factors? Are they receptor-based factors? And all of these things are what we call biomarkers. And they are what we, as an industry, are really, really interested in, because if we can identify the patient group that is going to be more successfully treated, then number one, we've made our drug development pathway much clearer, but we've also got much more opportunity to have a dramatic impact in those patients, which is, after all, what we're interested in.
Wonderful. So there are no more technical questions coming in. Perhaps I could ask one about careers and the practice of science in different ways. Your career was first, as many of us, academic, and that you headed an academic group, in immunology and skin biology, up until nearly a little bit short of 20 years ago, before coming across to industry. What do you see the differences or similarities between the practice of science in those two settings?
Wow, that's a really good question. So all I can tell you is my experience, and I think what I experienced when I went into industry was that people didn't work on their own. First of all, it was never success was not dependent on me as an individual having a good idea and being able to demonstrate through research whether it was true or not. Within pharma, it's all about working as teams. And why is it about working as teams? Because you have to demonstrate so many different things. They talk about it taking a village. Well, I think it probably takes a city, to get a drug from that research stage all the way to market. You need lots of people with different skills. So, they've got to have marketing skills. You've got to find out where your drug's going to be successful. You've got to have management skills and, scientific skills, which is, just about all I had when I went and joined Covance, were good. But in order to be able to interact with drug development, I had to understand the purpose of regulations, the questions that we were trying to answer. So it's much more focused, in what we're trying to deliver. And there were a variety of different analytics, different techniques that you could do. So you could answer the questions in lots of different ways. And that's part of the pleasure of it, is trying to be as accurate and precise as you can to give the regulators information that leaves them in no doubt of the question that you're trying to answer. So it was building this variety of different skills, which for me was great. I really enjoyed learning new stuff, but the difference from going from a relative expert in a field to that's great, but you need all of this other stuff as well. That was a massive learning curve. Yeah.
So if you had advice for somebody at a fairly early stage of their career, they're coming toward the end of their PhD training or a first postdoc, who were thinking about industry, how would you advise them to prepare for that?
Be prepared to rely on additional skills. Being a fantastic scientist is not enough. You have to be able to work in a team. You have to be able to enjoy being part of a team and working with people who have got have different skill sets, and and rely on those skill sets. There are people who are so much better organizers and managers, and it's that reliance on one another, which is really important and difficult to explain until you're in it. So, you know, if I'd just done my PhD and I was thinking of going into industry, it's recognition of you're going to be on a learning curve. Regulations are, the overarching understanding of what you're trying to do is super important. And science will get you part of the way, but not all the way. Yeah.
Wonderful. Well, thank you so much, Siân. I haven't any more questions myself or from the audience. I just want to check there's no further updates. So I'll bring matters to a close if I can. So can I thank all of you delegates for attending the webinar live. It's been a pleasure having you with us, and thank you for your questions. Thank you particularly to Siân for a really stimulating talk, canter through the world that we're all interested in. You can continue this conversation through Twitter if that is your practice. Please use the handles @BiochemSoc and @PP and then that conversation will be picked up. If you are looking for career changes and want more careers advice, the Biochemical Society has got facilities that can help you there. They have got, on their website, they've got career profiles, they've got advice on job seeking, and as we were discussing right at the end, there is quite a lot more than your science that you need to be thinking about as you try and develop your career. I'd like to encourage you all to think about the Biochemical Society and to become a member. These are new and very different times for all of us, and it's more important than ever to stay connected with one another. Joining the Biochemical Society is one very good way of staying in touch with this community. It's researchers from undergraduate level through to retirement, specialists in all fields of biochemistry, and membership is highly affordable and comes with a raft of benefits. Reduced fees for meetings, specialist events, courses, grants, bursaries, and personal subscriptions to some of the journals published by PP Publishing. So please visit the Biochemical Society website if that is of interest. It is biochemistry.org. I said at the start that you might be interested in nominating a topic of this type. Oh, sorry, I've missed a point. You might be interested in a conference that took place a year ago but was fortunately filmed. So that is a drug repurposing conference. You can watch on demand from Monday the 5th of October if those discussions will interest you. We'd be delighted to hear your suggestions for other events for the future Biochemistry Focus webinar series. You can feed those in through a process at the Biochemical Society, all of which is explained on their website, biochemistry.org/webinars. We want your ideas as what for topics or for speakers to illustrate those topics. And lastly, I'd like to tell you of an upcoming webinar to coincide with Biology Week, which is only a couple of weeks away, and the 10th anniversary of the Royal Society of Biology. The session is entitled "A Decade in Biology, What Have the Biosciences Done for Us?" and it will take place on Wednesday the 7th of October at 3 p.m. through to 4:30 p.m. British Summer Time. So there'll be 4 speakers at that who are authors in illustrating different areas of bioscience and what have been achieved in the last decade. So once again, I thank Siân Estdale for a really fantastic webinar, and the Biochemical Society and Portland Press for hosting this event. And thank you very much.